We were previously talking about female infertility. We defined infertility as the incapability to conceive after 1 year of unprotected intercourse with the same partner. These all criteria are required for the definition of infertility. So we say there should be no use of contraceptives or interruptions. There should be the frequency of intercourse should be optimal, and the it should be with the same partner, and the uh, intercourse should be longer, like up to 1 year. And the why it is needed for 1 year, because the ability to conceive is not 100% in a single cycle of the female. So apparently healthy couples, they conceive only 20% of them conceive in within 1 month of the cycle, and about 60 to 70% they would conceive in 6 months, and in 1 year about 85 to 90% couples they conceive, and the rest, that are the 15 to 10 to 15%, they would be now called as infertile because they could not conceive within 1 year of intercourse. So the probability of conceiving in one menstrual cycle is called as fecundity of a couple or a woman. So we start evaluation of the female infertility or infertility of the couple after 1 year, but we can do it before it, because the if there are previous history of infertility in uh, both or one of the partner, or there could be the increasing age of the female, could be other hormonal issues, PCOS, polycystic ovarian syndrome. So we can start it at 6 months, the evaluation at 6 months, but mostly they are encouraged after 1 year of the unprotected intercourse if there is no conception. So last time we said that female infertility contributes about 40 to 50% of the infertile couples. So these causes of female infertility could be due to they could be ovulatory, they could be tubal, they could be uterine, they could be cervical. So if we see ovulatory causes of infertility, they could be ovulatory dysfunctions. So these ovulatory dysfunctions, they are divided into 7 categories. So the first one, the group 1, is hypothalamus pituitary axis failure, and we know the important one is Kallmann syndrome, but it is rare. Then there is anorexia nervosa. It is associated with the hypothalamus pituitary axis failure. Anorexia nervosa mean decrease in appetite, and there is tendency to appear as a lean. So then in group 2, hypothalamus pituitary axis dysfunction, and it is due to hypothalamus pituitary axis failure, mean the hypothalamus and pituitary, they are not producing GnRH, and in a hypothalamus pituitary axis dysfunction there could be uh, uh, dysfunction at any level, like there could be dysfunction of feedback or anything. So it could be hypothalamus pituitary axis failure, we can say, is the secondary cause of infertility in case of females, and hypothalamus pituitary axis dysfunction would be like primary cause. We say primary cause, that is ovarian cause, and the secondary cause, that is other than ovarian cause. So in case of hypothalamus pituitary axis dysfunction, there would be dysfunction of feedback like mechanism, we'll see it later. So it is the diseases that are present would be polycystic ovarian syndrome, could be hypothyroidism, could be then there could be group 3, and that is ovarian failure. that ovaries, they are not producing or they are not ovulating, so it would be early ovarian failure or premature ovarian failure. And then in group 4, there could be genital tract malformation or alteration, so they are Asherman syndrome, we'll see it like later. And then group 5, prolactinomas, high prolactin production, so there are tumors that are in pituitary. We know pituitary is releasing prolactin, so if there are tumors, then a high concentration of prolactin would be present in the blood, and we know the prolactin, it inhibits gonadotropin releasing hormone and gonadotropin release, so there would be infertility in females. And then there could be group 6, that is functional hyperprolactinemia. Functional hyperprolactinemia mean that if a mother is lactating, then the prolactin would be given feedback of the suckling, so the prolactin levels, they would be high than normal in non-lactating state. So these high prolactin that are called as functional hyperprolactinemia, they are responsible for decrease in the gonadotropin and GnRH. And then there is group 7, hypothalamus pituitary tumors are non-prolactin secreting tumors, tumor present in the non in other than prolactin secreting sites, and they are not secreting hormones, so there would be, they are called as pituitary adenomas. Adenoma mean it is in the anterior part of the pituitary. So these are the 7 groups, we'll see them later one by one. So before going to the details of the ovulatory dysfunctions, we should know about the normal ovulatory function of the ovaries. The most important feature of the female reproductive tract is its cyclicity. The cyclicity are the periodic changes in the female reproductive tract, unlike the men or the male reproductive tract. And these changes, they are governed by the hormonal changes. So they help to prepare the female reproductive tract for fertilization and pregnancy. In primates and human, this cycle is called as menstrual cycle, and the most common feature, the most important feature of the menstrual cycle is the periodic vaginal bleeding that occurs with the shedding of uterine mucosa, and that is also called as menstruation. In case of said menstrual cycle is actually a cyclic activity that is present at two levels. It is present at ovarian level as well as uterine levels. So the ovarian part, it is having regular cyclic growth of follicles and then release of an ovum each month. And the ovarian cycle, it would be we can say made up of two phases: the proliferative phase, that is the growth phase, and then is secretory phase, release of a lot of hormones, LH and estrogen. And then there would be in uterine cycle, it is the uterine part, and it is in concordance to ovarian cycle. It is again regular cyclic growth of endometrium, the internal lining of the uterus, and then shedding when there is no pregnancy. And it could be maintained if there is pregnancy by a corpus luteum. So it is again made up of two phases: proliferative phase and secretory phase. So these phases, they superimposed on each other. The ovarian cycle phases and the uterine cycle phases, they overlap each other. They are coinciding, they are helping each other. So the in order to understand ovarian cycle, we have to know about the hormones first that are being released from the axis that is called as hypothalamo-pituitary ovarian axis. So hypothalamo-pituitary ovarian axis is a cascade of the gonads, these glands being regulating and being regulated. So the hypothalamus we know is part of brain, and it is the controlling pituitary gland. So hypothalamus we know it has nuclei, the group of neurons that are cell bodies of neurons that are releasing factors. So arcuate nucleus and preoptic nucleus, they are releasing gonadotropin, releasing the hormone from hypothalamus, and then these gonadotropin releasing hormone, they are coming to pituitary where there are specialized type of cells and they are called as gonadotropes. These gonadotropes, they would release gonadotropin. Gonadotropin are follicle stimulating hormone or FSH, and luteinizing hormone or LH. And then these FSH and LH, they would go to ovaries through blood, so they are endocrine. And then in ovaries, they would release estrogen, progesterone, and inhibin. So estrogen, progesterone, and inhibin, they would come back and they would give negative feedback to the pituitary, and they would give the negative feedback to hypothalamus in order to decrease the level of follicle stimulating hormone and luteinizing hormone. We have to keep it remember that progesterone and inhibin, they always give negative feedback to the pituitary and hypothalamus. But estrogen, when its levels become very high, the surges of estrogen, they come near an ovulation, they would give positive feedback to pituitary in order to release more LH in order to bring ovulation. So we'll explain it later. But we have to keep it remember that estrogen in normal levels, in low levels, it gives negative feedback, but in high levels, it would give positive feedback to pituitary in order to release more LH. Next, we have to understand where these hormones, LH and FSH, luteinizing hormone and follicle stimulating hormone in ovaries, they work. In ovary, there is a lot of stroma and then there are follicles. Stroma, stroma is a mesenchymal type of tissue, and the follicles, they are, for example, this large Graafian follicle, it is made up of the ovum that is present inside, and then there are cells, cuboidal cells. These are granulosa cells. There are many layers of these granulosa cells, and then there are theca cells, layers of theca cells around the granulosa cells. So these theca cells, they would be theca interna and theca externa. Two layers are present over there. So these theca cells, they have LH receptors. We have to keep it remember that when Graafian follicle is growing, then theca cells, they have LH receptors, and the granulosa cells, they only have FSH receptor or follicle stimulating hormone receptor. So theca cells, they are present on outer side. They are also vascular layer of the follicle. Mean vascular mean blood vessels, they come up to the level of theca layer. They won't go into the granulosa cells or granulosa layer. In granulosa layer, they are avascular, mean blood vessels are not present. So the hormones, they are either LH or FSH, they are coming at theca cell first because they are coming through blood vessels in blood. So the LH, it can directly bind to the receptors on the cell membrane of theca cell. We know LH is protein hormone. It is globule, it is glycoprotein. It would not go into membrane inside. It has to bind on, it cannot cross membrane, it has to bind on the membrane. So its receptors are present on the membrane of theca cell. So they would bind with their receptors, and then they help the cholesterol to get absorbed and would be converted into the androgens. The first one is androstenedione. So that is a type of androgen that is produced in theca cells. and then this androstenedione, it would cross the membrane of theca cells and would go into the they can directly cross membranes because they are cholesterol derivatives, they are lipid derivatives, so they would go into the granulosa cells. In granulosa cells, when FSH would come and bind with the granulosa cells, and it is again seeping from theca cell because granulosa is avascular, so blood vessel they have released LH and FSH both on the theca cells, and then FSH has come up to the level of granulosa cells, so it would bind with the its receptor on the membrane of granulosa cell, and then it would start a cascade of reaction that would help aromatase enzyme to aromatize the androstenedione into estrone, that is the estrogen 1, and then it would be converted into estradiol, or that is called as E2. So this estradiol is the estrogen that we commonly known as estrogen. So estrogen we know is a bigger class, so estrone is one of the member, and then it is converted into estradiol. So it means the hormones are being converted uh, when LH is present. Cholesterol is absorbed into theca cells, then this cholesterol is converted into androgens. Androgens are seeping into granulosa cells, and in granulosa cell their aromatization is occurring if there is FSH. If there is no FSH, then aromatization cannot be taken place. Similarly, if there is no LH, then the androgens could not be formed and estradiol could not be formed. It means both hormones are necessary for the production of hormones. This is called as two-cell, two-gonadotropin hypothesis. Mean there are two cells, theca cells and granulosa cells, and then there are two gonadotropin, that is LH and FSH. They are acting on these two different types of cells. So it is called as two-cell, two-gonadotropin hypothesis. Ovarian cycle of the follicular growth. You can see here at the first stage there are primordial follicles, and there are a lot of primordial follicles, and then these primordial follicles, they would be converted into primary follicles, and then primary follicles would be converted into the third and fourth stage, that is pre-antral follicles, before the appearance of a cavity in the follicles, and then after that there are antral follicles, and then there would be ovulation. So before ovulation, this whole growth of the follicles, it is supported by FSH. So FSH is for the growth of follicles, so that the size of the follicles, it would become up to 20 to 24 millimeter. That would be called as Graafian follicle. So 20 to 24 millimeter uh, size of the diameter of the follicle would be called as Graafian follicle. Up to that level, it is supported by FSH. So we can differentiate this, that primordial cells, they have grown uh, 70 days without the use of FSH or LH, and they are under the influence of growth factors. After that, it is supported by FSH most of the time, but the final growth of the Graafian follicle is supported by LH also, and we can say final maturation. So then there is ovulation. So this ovulation is supported by LH. High peaks of LH comes, and they are responsible for the release of ovum from the granulosa and the theca cells. That would be called as ovulation. So ovulation is actually an inflammatory process. So there would be formation of, after ovulation, there would be formation of corpus luteum, and the granulosa cell, we know they have only receptors uh, of FSH previously. Now they get receptors of LH also, so that is called as luteinization of the granulosa cell so that they can respond to LH and they can release progesterone. So after formation of corpus luteum, previously the granulosa cells, they had only the receptors for FSH, but now after ovulation, here the remaining granulosa cells, they have receptor for LH also. So that is called as luteinization of the granulosa cell. Luteinization means they have grown receptor, they have appeared receptors for luteinizing hormone, so that LH can act on them directly and produce progesterone out of them. This whole cycle of the follicular growth and ovulation and the formation of corpus luteum and then disintegration of corpus luteum, this whole cycle, it would take about 28 days. So these half of this these 28 days is supported by, that is 14 days, they are supported by most of the time by FSH, and half is supported by LH. So these first half is called as the proliferative phase, and the next half is called as ovulatory phase. So these two phases, they are superimposed on lutein cycle. We'll see it later. As we said previously that the ovarian cycle superimposed on the lutein cycle. Ovarian cycle we saw is made up of proliferative phase or pre-ovulatory phase, and then is the luteal phase or the post-ovulatory phase. Same is the case with the uterine cycle. It is made up of two phases, the pre-ovulatory or proliferative phase, that is coinciding with the ovarian proliferative phase or pre-ovulatory phase, and then there is a post-ovulatory phase in uterine cycle that is coinciding with the post-ovulatory or luteal phase of the ovaries. So if we know the normal menstrual cycle of the women is 28 days, so we start counting the proliferative phase from the first day of bleeding or menses, start of menses, that would be the first day. So up to first to 5 days there would be bleeding, that is the destruction of endometrium. We know endometrium is the internal most lining of the uterus, and it is made up of two layers, basal layer and the functional layer. So functional layer, it start to shed at the first day, and then it is we know it is made up of lot of glands. So this functional layer, it would shed between 1 to 5 days of the menstrual cycle, and then there would be start of the regrow of this functional layer under the influence of the estrogen that is being released from the Graafian follicle that has formed till that time up to 5 days. So this Graafian follicle, it has granulosa cell that are releasing a lot of estrogen, and this estrogen start to build endometrium of the functional endometrium of the uterus, and then it keeps on proliferating or building up to the day of ovulation. So after ovulation, we know in ovaries there would be remaining theca cells and granulosa cells. These cells, they would start to produce progesterone under the influence of LH, and this progesterone, it helps the post-ovulatory phase of uterine cycle, or the it is also characterized by the secretory changes in uterine endometrium. So this progesterone, it helps the uterine endometrium after ovulation to start secreting. It would become the glands that are present in the endometrium, they would become more elaborated. So if we see histology of the endometrium before ovulation, and after ovulation, or we can say in proliferative phase and in the secretory phase, there are marked differences. For example, in case of the endometrium before ovulation, the glands, they are narrow, their lumens are narrow, and they are simple tubular glands that are lined with a layer of columnar epithelial cells. And after ovulation, the progesterone, it would help these glands to become enormous, and they, if there was previously proliferation, now there would be secretory changes. So these glands, they would, their lumens would become broadened, and their cells, these glands, they would appear like corkscrew shaped, and they would become like spiral like, and they become more secretory. There are two marked changes in these glands, and most of these changes, they would occur after the first half of the secretory phase of the uterine cycle, in, or we can say, last phase of the uterine secretory cycle. So in this phase, there is appearance of vacuoles that are filled with glycogen under nucleus of these columnar cells. So these glycogen-filled vacuoles, they are called as subnuclear vacuoles, or it is called as subvacuolar vacuolation. And these subnuclear vacuoles, they help the early uh, embryo to grow if there would be pregnancy. So, because they are filled with glycogen, so they will nourish embryo. And the second change that would occur is that these glands, they would now become like corkscrew shape. So these are the two main changes, and then there is another change that is called as leukocytic infiltration. So there would be a lot of WBCs in the endometrium. They would enter from blood vessel into the endometrium. If there is corpus luteum, it stays, then there would be stay of the secretory phase of the endometrium. And we know, if there is no pregnancy, corpus luteum, it has fixed life, that is of 10 to 12 days. So there would be luteolysis if there is no pregnancy. Luteolysis means disintegration of corpus luteum, and this corpus luteum, it undergoes apoptosis, the normal cell death process. We know if there is pregnancy, then this corpus luteum is supported by human chorionic gonadal often from placental cells. So if there is no pregnancy, it means there is no placenta, and there would be death of corpus luteum, and that is called as luteolysis. So when there is luteolysis, there would be no progesterone. Progesterone would dip down, and after, when the progesterone would go down, because there is no corpus luteum, this uh, secretory uterine endometrium, it would be disintegrated, because there would be production of a lot of cytokines, prostaglandin, and inflammatory mediators. And these inflammatory mediators, prostaglandin, cytokines, they would help the endometrium to disintegrate, and they would do enzymatic digestion of the endometrium, and there would be shed of the endometrium. If we see structure of the endometrium, it is made up of these layers. We can see here myometrium, that are the muscles, and then is the endometrium, that is made up of functional layer, the layer that is open toward uterine lumen, and then there is a basal layer. So this basal layer, it won't go to disintegration, and these layers of the endometrium, they are supplied by various blood vessels like radial branch or radial artery. It is supplying the basal layer of the endometrium, and spiral arteries, they are supplying the functional layer of the endometrium. So we can see in basal layer there are bases of the glands, and these glands, they are opening toward lumen, and the most of the gland, the part of the gland is present in the functional layer. So when there is withdrawal of progesterone, when there is no corpus luteum after 10 or 12 days, and when there is no pregnancy, it means there would be shed off of this functional layer. This functional layer of the endometrium, it would go off, and when it would go off, there would be bleeding that would stays for 4 to 5 days, and it is very moderate type of bleeding. So what stops this bleeding? So we know when there would be loss of this layer, the functional layer, there would be loss of these blood vessels that are the spiral blood vessel here, and then the remaining spiral blood vessel, they would undergo vasoconstriction. So the stoppage of blood is like happening in normal other parts of the body. So there is vasoconstriction when there is damage to blood vessel, they would constrict, so there would be no loss of blood vessels, so blood. And then there would be clotting mechanism, the normal clotting mechanism. We know there are a lot of clotting factors circulating in the blood, and they with the help of platelets, they would make a clot. We know the fibrinogen, there are the prothrombin, and so on. So then, so the second step is, the first is vasoconstriction that happens in other parts of the body also when there is a damage to the blood vessel, and then there is the normal clotting process, and then there would be re-epithelialization of endometrium. So there would be formation of new functional layer of the epithelium. So it start to form after 5 days of the bleeding. Just after 5 days, because bleeding stops because the vessels, they have constricted, the spiral vessels, arteries, they have constricted. There was clotting process, and then there was reformation of this functional layer started. So the bleeding stops due to these three processes. We know the menstrual cycle, it is of 28 days, and it is perfectly timed event. It is very regular, and we know the regularization of menstrual cycle is only done by ovulatory function or ovulatory process. So at the start of the follicular phase in ovaries, that is the day one, there are a lot of primordial follicles uh, present in ovary, and we know the females, they have the finite number of primordial follicles. So these primordial follicles at day one, they already have undergone 70 days of maturation or growth, and this uh, growth was independent of LH and FSH. It was only done by various growth factors, and after that, these primordial follicles, if they won't get FSH, they would die. They would not grow, and so there is requirement of peak of FSH. So they start the increasing FSH from the pituitary. It start to release, and it comes into the blood and act on these primordial follicles to grow them, and then we know they would pass to antral phase, pre-antral, and then the antral phase, and then there is start of release of due to action of FSH, there is start of release of estrogen and inhibin beta. So this inhibin B R and estrogen, they would act on pituitary and give negative feedback. So this FSH, it would help these primordial follicles to grow one uh, dominant follicle that would ends up in Graafian follicle. So at the time of formation of Graafian follicle, there is decrease in FSH. This decrease here we can see here. FSH is decreasing when there is formation of antral follicle or the dominant follicle because of the negative feedback by estrogen, increasing estrogen, and the inhibin B on the pituitary. So estrogen and the uh, inhibin B, they were increasing. Here we can see the peaks of or surges of estrogen, and they have decreased FSH because the function of FSH is almost completed. So FSH is decreasing. The dominant follicle, it has most receptors for the FSH, so it would grow uh, faster than the others. So the other, because they don't have FSH receptors, so they would die, and only the Graafian follicle or the dominant follicle, it would stay, and it would be releasing estrogen and inhibin. So the dominant follicle, it or Graafian follicle, it releases estrogen in high amount, a lot of estrogen, and there would be a peak of estrogen in the blood. And this peak of estrogen, previously the lower levels of estrogen were giving negative feedback on the pituitary, but the peaks or high levels of estrogen, they would give positive feedback to pituitary to release more LH from the pituitary. So there we can see previously the LH was, the levels of LH were stationary, they were in a continuous form, and now there would be a peak of LH when there is a peak of estrogen. Why there is peak of estrogen? Because Graafian follicle, it is releasing a lot of estrogen. So this estrogen, it gives positive feedback on pituitary to release more LH. So there would be a peak of LH, and this LH, it would act on the thecal cells, and it would bring inflammatory process, that is formation of cytokines and the inflammation mediators. So there would be release of ovum from the uh, follicle, that would be called as ovulation. After ovulation, we can see there is formation of corpus luteum. So this corpus luteum, it start to release progesterone and inhibin alpha. So or inhibin A. So inhibin A or inhibin B, they both have function of negative feedback mechanism. So progesterone and the so there would be at ovulation, we can see here peak of or increasing levels of progesterone. So these progesterone, they are coming from corpus luteum with the inhibin A. So they are giving negative feedback to the pituitary to not release more LH and FSH. So after 8 days of ovulation, the progesterone, it reaches to its peak. So there would be decrease in LH at the that time. So LH is given negative feedback and it has decreased. So uh, during formation of corpus luteum or during luteal phase, the not only progesterone but estrogens are also released, so they are going hand in hand. If there is pregnancy, then human chorionic gonadotropin, it would sustain this corpus luteum to keep on releasing the high levels of progesterone, and if there is no pregnancy, then this corpus luteum, it would disintegrate, it would be undergone luteolysis, and after that there would be no progesterone, there would be no negative feedback on the pituitary, so there would be release of FSH again. So there would be again start of the formation of or growth of these primordial follicles to undergo into the process of formation of selection of one dominant follicle for the release of ovum, or there would be ovulation. So this cycle again started due to uh, that is the submission of the progesterone. Progesterone would go down due to the disintegration of corpus luteum, and then LH and FSH that were being given negative feedback due to this progesterone and inhibin A, they would uh, come out of that negative feedback, and then FSH levels, they would be increased, and then there would be start of cycle again. What tells the pituitary to release FSH, and then what tells it to release LH? It depends on the release of GnRH. So it is known that the GnRH, it is being released in pulsatile manner. So for LH, first surges of GnRH would come, and there would be release of a lot of LH. And for FSH, there are slow surges of the slow waves of the GnRH, they would come. So there is another hypothesis that GnRH is in two form. It is LHRH, that is luteinizing hormone releasing hormone. It could be follicular stimulating hormone releasing hormone. But there are no concrete evidences for this hypothesis. So we only depend on these pulsatile frequency of GnRH. So for fast pulsatile frequency, there would be release of LH. For slow, there would be release of FSH. So at the start of cycle, there would be slow pulses of the GnRH, and at the uh, ovulation, there would be pulses of GnRH that are faster. So that is all about menstrual cycle.